Antibodies, also known as immunoglobulins, or Ig, are key players in the humoral immune response, circulating in body fluids to help neutralize and eliminate pathogens. Antibodies are proteins made by B cells. When B cells become plasma cells, they turn into antibody factories, pumping out large amounts to fight off invaders. Antibodies specifically bind to antigens, which are any substances that trigger an immune response. This binding can neutralize pathogens, tag them for destruction, or activate the complement system to help eliminate them.
Antibodies circulate in the blood, lymph, and other body fluids, providing both systemic and local defense to protect the body from pathogens wherever they appear. Antibodies have a distinctive Y-shaped structure. The Fab region at the tips binds to the antigen, while the Fc region at the stem connects with immune cells to help coordinate the response. Antibodies can neutralize toxins and viruses. They do this through neutralization, binding to toxins or viral particles to block their activity or prevent them from entering host cells. Antibodies can also tag pathogens for destruction, a process called opsonization. Basically, they coat the invaders, making it easier for phagocytes like macrophages and neutrophils to grab and destroy them. Antibodies can make pathogens clump together, a process called agglutination. By cross-linking multiple invaders, they form clumps that are much easier for phagocytes to engulf and remove. Antibodies can also precipitate soluble antigens. They bind to these antigens to form insoluble complexes, which phagocytes can easily remove. This is especially important for detoxifying circulating toxins and microbial products. There are 5 main classes of antibodies: IgG, IgA, IgM, IgE, and IgD, each with its own role in protecting the body from infections. IgG is the most common antibody in humans, making up about 70 to 75% of all serum antibodies. It's responsible for long-term immunity and memory responses, helping the body remember past infections. IgG has 4 subclasses: IgG1, IgG2, IgG3, and IgG4. They mainly differ in their hinge regions and disulfide bonds, which slightly change how they function in the immune response. The hinge region of IgG is like a flexible joint. It lets the Fab arms move to bind multiple antigens, and also affects how well IgG can activate complement and form immune complexes. Among the IgG subclasses, IgG3 has the largest hinge region, giving it extra flexibility to bind antigens. It's also the most powerful activator of the classical complement pathway, making it highly effective at helping clear infections. IgG2 and IgG4 are poor complement activators. IgG2 mainly responds to polysaccharide antigens and has weak complement activity, while IgG4 is involved in chronic antigen exposure and immune tolerance and doesn't efficiently trigger complement at all. All IgG subclasses can cross the placenta except IgG2. IgG1, IgG3, and IgG4 pass through efficiently, giving the fetus passive immunity, while IgG2 is transported poorly, offering less protection against polysaccharide encapsulated bacteria. IgG is involved in the secondary immune response, which is much faster and stronger than the first encounter. It's the main antibody produced when the body meets a previously recognized pathogen. IgG1 and IgG3 mainly respond to protein antigens, such as toxins and viral proteins. These 2 subclasses are especially dominant when the immune system is dealing with protein-based threats. Macrophages, monocytes, and neutrophils have special Fc receptors for IgG. When IgG binds to these receptors, it makes phagocytosis much more efficient. These receptors, called Fc gamma receptors, Fc gamma R, recognize the Fc portion of IgG, helping immune cells grab and destroy tagged pathogens. IgG has a high diffusion capacity, so you can find it almost equally inside blood vessels and in surrounding tissues. Because it's small and flexible, it can easily move out of the bloodstream to reach sites of infection. IgG is better at precipitation than agglutination. Precipitation involves small soluble antigens, while agglutination involves large particles like RBCS. IgM is more efficient at agglutination than IgG.
IgM is called a macroglobulin because of its large size. It makes up about 5 to 10% of all serum antibodies. IgM has two forms. In the serum, it's a pentamer, making it great at agglutination and activating complement. On the surface of B cells, it's a monomer and acts as a B cell receptor, BCR, to recognize antigens. IgM is made of 5 monomers linked together by a J chain, a special glycoprotein. The J chain helps stabilize the pentamer and makes it easier to secrete IgM at mucosal surfaces. IgM has a star-like shape with 10 antigen binding sites, making it extremely effective at binding multiple antigens at once. IgM mostly stays in the blood vessels because of its large pentameric size. Unlike IgG, it's too big to cross the placenta, so it doesn't provide fetal immunity. IgM is the first antibody to appear after an infection, making it the primary responder in the immune system's defense.
In newborns, IgM is the first antibody they can make themselves, while IgG comes from the mother through the placenta to provide early protection. IgM is a primary response antibody, and its production only continues while the antigen is present. Once the infection is cleared, IgM levels drop. When you detect IgM, it usually means an acute or recent infection. IgM appears within days after infection and then declines over the next few weeks, so in serology, its presence points to a current or recent infection, not long-term immunity. IgM is the most efficient antibody at activating the classical complement pathway. In fact, just one IgM pentamer bound to an antigen is enough to activate C1. That's why ImG is especially powerful against bacteria, particularly during the early, acute phase of infection. In the bloodstream, IgA is found mainly as a monomer, circulating to help defend against infections. IgA has 2 subclasses, IgA1 and IgA2. They mainly differ in their hinge region, which is about 22 amino acids long and affects how the antibody functions. IgA2 has a shorter hinge region compared to IgA1, which makes it more resistant to bacterial proteases and better suited to defend against pathogens at mucosal surfaces. IgA1 is found mainly in the serum. It has a longer hinge region, which makes it flexible but more vulnerable to bacterial proteases. IgA2, on the other hand, is found mostly in mucosal secretions like saliva, tears, intestinal fluid, and breast milk. Its shorter hinge region makes it resistant to bacterial proteases. Serum IgA mainly acts as an anti-inflammatory antibody, unlike secretory IgA, which blocks pathogens from sticking to mucosal surfaces. It can downregulate IgG-mediated responses, including phagocytosis and cytokine release, helping prevent excessive inflammation. Secretory IgA, SIgA, is mostly found as a dimer at mucosal surfaces. You can find it in saliva, tears, sweat, colostrum, and breast milk, where it helps protect the body from invading pathogens. IgA is mainly produced in MALT. That's the mucosa-associated lymphoid tissue, including the tonsils, Peyer's patches, appendix, and other mucosal sites. B cells in MALT become plasma cells that secrete dimeric IgA to protect mucosal surfaces. The body produces a lot of IgA every day, second only to IgG. Most of it is secreted across mucosal surfaces as dimeric secretory IgA, which is why serum IgA levels are relatively low and mainly monomeric. Secretory IgA is a dimer held together by a J chain, and it has an extra part called the secretory component. This secretory component comes from epithelial cells, specifically from the polymeric IgG receptor. Its job is to protect IgA from enzymatic digestion and help it survive the harsh conditions at mucosal surfaces. Importantly, only secretory IgA has this component. Serum IgA does not. IgA in breast milk, especially in colostrum, provides passive immunity to newborns, helping protect their mucosal surfaces during early life. IgD was first discovered in a patient with multiple myeloma, which helped scientists identify it as a distinct class of immunoglobulin. IgD is the least abundant antibody in the blood and is rarely found circulating. Instead, it's mainly located on the surface of naive B cells, where it works together with IgM as a B cell receptor to help recognize antigens. The exact function of IgD isn't fully understood, but it seems to play a role in B cell activation and regulation, helping the immune system respond appropriately to antigens. IgE binds to immune cells through its Fc region. On mast cells, it triggers degranulation during allergies. On basophils, it releases histamine and other mediators. On eosinophils, it helps fight parasites. And on Langerhans cells, it aids in presenting antigens. Overall, IgE is key for allergic reactions and defending against helminths. That would be all. Thank you for watching.